So coming to the result and discussion section, let us first understand about the morphology or macroscopy. So remember, my dear students, both macroscopy and morphology are synonym with each other. This Cassia bark is very easy to identify because it is having its characteristic aroma due to the presence of the volatile oil in it. Moreover, the shape of Cassia bark, either it is a single quill or channel piece. As in the diagram you can see, the both a single quill and channel pieces are available. So coming to the organoleptic character, it is dark brown in color, odor sweet and aromatic, taste sweet and slightly mucilaginous. Coming to some extra features, adhering cork is grayish in color. So outermost layer, the raw bark when collected, the outermost layer is grayish in color. It is due to the presence of adhering cork. The fracture means if we break it, then it is easily breakable. It is short and granular. So shape already I mentioned, it is either single quill or channel piece. Coming to the length, so length obviously depends on the method of collection, but in general Cassia bark is 5 to 40 centimeter in length, whereas 1 to 2 centimeter in its diameter. So inner surface is a little bit reddish in color. It is due to the presence of tannin in it, very minimum quantity of tannin in it, whereas the cracks, transverse cracks may also be observed in the Cassia bark outermost layer. So these are the various macroscopical or morphological features of the Cassia bark. Coming to its microscopy, so let us see one by one. So when we will take the transverse section of Cassia bark under the microscope, you will observe the cellular characters exactly like denoted in this figure. So the topmost part is known as the cork. So any bark, if we take the transverse section, obviously it will be consisting of the cork, followed by 2 to 3 layers of cell known as phellogen and phelloderm. Both phellogen and phelloderm are merged with each other, hence it is not distinctive. Then followed by, you will find the scleroid cell. It is present individually or in a group. Now below to that, this part is known as the cortex region. You will find a very distinct pink colored zone upon staining with fluoroglucinol and HCl 1:1 ratio. A pink color band, what is we have mentioned here, this is known as sclerenchymatous band or stone cell layer band. So what is stone cell? It is a parenchyma cell which is brick shape with U-shaped thickening. Its shape is brick shape with U-shaped thickening. It consists of lignin. Due to the presence of lignin, when we will stain with fluoroglucinol and HCl 1:1 ratio, it gives pink color. So the entire region, sclerenchymatous band, will appear pinkish color. This is one of the important identification feature of Cassia bark. Now moreover, you will also observe the pericyclic fiber. These are also lignified character in it. Next, you will observe very distinct oil glands, which is yellowish color in nature. So these oil glands contain volatile oil, due to which Cassia bark possesses its characteristic aroma. Now medullary rays, 3 to 4 layers of cells, which is horizontally arranged, which is vertically arranged. So 2 to 3 layers of cells are present in it. So in the transverse section, if we add iodine solution, then few very minimum quantity of starch grains are also observed in the inside the cells. So these are in brief various microscopical characters are found in the transverse section of Cassia bark. Cork followed by phellogen phelloderm, scleroid cell and sclerenchymatous band, which is an important character, pericyclic fiber, medullary rays, then oil glands and starch grains. So these are the various microscopical features present in Cassia bark.